Hi everybody, I'm Dr. Biomed. Welcome to the Biomed Cafe channel. In this channel, complex biological and medical contents are presented in a simple, classified and summarized form. Also, I have designed a quiz game for you. At the end of the video, I will explain the interesting quiz game. The topic of this video is bulk transport across the cell membrane. Please subscribe to this channel, and if you like the contents of this video, please like it. Bulk transport. It's an energy-intensive process. Material transported by bulk transport are large particles and large quantities of smaller particles. Function of bulk transport. It allows cells to obtain nutrients from the environment. It selectively grabs certain particles out of the extracellular fluid, and it releases signaling molecule to communicate with neighbors. Bulk transport can be divided into two subgroups: endocytosis and exocytosis. Endocytosis. Endo means internal and cytosis means transport mechanism. It's a type of active transport that moves particles into a cell. Material transported by endocytosis are large molecules, parts of cells, and whole cells. Now I want to talk about the steps of endocytosis. As you see, the plasma membrane folds inwards or invaginates. Then a cavity full of extracellular fluid and particles is formed. The plasma membrane folds back on itself until the ends of the infolded membrane meet. Then this traps the fluid inside the vesicle. The vesicle is pinched off from the membrane as the ends of the enfolded membrane fuse together. Finally, the internalized vesicle is then processed by the cell. You can see here all of the endocytosis steps together. Endocytosis can be divided into three subgroups: Phagocytosis, Pinocytosis, and receptor-mediated endocytosis. Phagocytosis. Phagocytosis or cell eating. It's a process by which large particles are taken in by a cell. Material transported by phagocytosis is microorganisms, apoptotic cells, larger macromolecules, whole cells, and cellular structures. Examples of phagocytes are neutrophils and macrophages. They eat invaders microorganisms through phagocytosis. Now I want to talk about phagocytosis steps. One. Detection. The phagocyte detects the antigen such as a bacterium and moves toward it. 2. Attachment. The phagocyte attaches to the bacterium and surrounds the bacterium. 3. Ingestion. The surrounded bacterium is enclosed within a vesicle. This vesicle with bacterium enclosed and internalized by a phagocyte. 4. Fusion. The phagosome fuses with a lysosome. And 5. Elimination. The release of digestive enzymes of lysosome degrades the bacterium. The degraded material is expelled from the cell by exocytosis. Pinocytosis. Pinocytosis or cell drinking. In pinocytosis, cells take in molecules which the cell needs from the extracellular fluid. Pinocytosis can be divided into two subgroups: Micropinocytosis and Macropinocytosis. Micropinocytosis. There are micropinocytic vesicles. These vesicles have 0.5 micrometers diameter. The shape of these vesicles is small and coated. They form by budding from the cell membrane, and they are located in most types of body cells. Material transported by micropinocytosis are liquids and water. The steps of micropinocytosis is like the endocytosis. You can see here the steps of micropinocytosis. As you see, these steps are like endocytosis, but there are coated vesicles here. Macropinocytosis. There are macropinocytic vesicles. These vesicles have 0.5 to 5 micrometers diameter. Their shape is large and uncoated. They form by ruffles that they are extended portions of the membrane. They are located in white blood cells, and material transported by macropinocytosis is extracellular material, such as soluble molecules, nutrients, and antigens. I want to explain about macropinocytosis steps. As you see, actin filament polymerizes at a cell membrane. Then it pushes the membrane forward into ruffles. It folds inwards and fuses with the basal membrane to form large membrane vesicles. And finally, the internalized vesicle is then processed by the cell. You can see all of the macropinocytosis steps together. The third subgroup of endocytosis is receptor-mediated endocytosis. It's a targeted variation of endocytosis. There are receptors in the plasma membrane that have binding affinity for certain ligand. And also there are clathrin proteins that attach to the cytoplasmic side of the plasma membrane. Material transported by receptor-mediated endocytosis is large quantities of macromolecules. I want to explain about receptor-mediated endocytosis steps. At first, the specified molecule or ligand binds to the receptor on the plasma membrane. After ligand-receptor complexes accumulate in the clathrin-coated pit, the region forms an invagination that is internalized by endocytosis. Then, a clathrin-coated vesicle is formed which encapsulates the ligand-receptor complex and extracellular fluid. The clathrin-coated vesicle fuses with an endosome in the cytoplasm and the clathrin coating is removed. And finally, lysosomal enzymes degrade the specified molecule and deliver the desired contents to the cytoplasm. You can see here all steps of receptor-mediated endocytosis together. Exocytosis. Exo means external and cytosis means transport mechanism. The reverse process of moving material into a cell is exocytosis. The purpose of exocytosis is to expel material from the cell into the extracellular fluid. Material transported by exocytosis are membrane proteins and lipids, soluble proteins, and waste material, microbes, and cellular debris. Exocytotic vesicles can be divided into three subgroups: secretory vesicles, transport vesicles, and waste vesicles. Secretory vesicles come from the Golgi apparatus and contain membrane proteins and lipids. Transport vesicles come from the Golgi apparatus, contain soluble proteins. Waste vesicles contain waste material. Exocytosis can be divided into three subgroups: constitutive exocytosis, regulated exocytosis, and lysosome-mediated exocytosis. Constitutive exocytosis. This action is performed by all cells. It delivers membrane proteins and lipids to the cell surface and to expel substances to the cell's exterior. It has complete fusion, and vesicle in constitutive exocytosis is transport vesicle. I want to talk about constitutive exocytosis steps. The first step is vesicle trafficking. The cell vesicles are transported to the cell membrane through the microtubules of the cytoskeleton. The second step is tethering. When the cell vesicles reach the cell membrane, there is pull towards the cell membrane and they eventually come into contact with each other. The third step is docking. This is the attachment of the vesicles with the cell membrane, initiating the merging of the phospholipids of the vesicle membrane with that of the cell membrane. And the final step is complete fusing. It involves the full fusion of the vesicle membrane with the lipid cell membrane, but the use of energy or ATP. You can see here all steps of constitutive exocytosis together. The second subgroup of exocytosis is regulated exocytosis. This mechanism depends on extracellular signaling for the release of its content onto the outer part of the cell. It involves the secretion of hormones, neurotransmitters, and digestive enzymes. Vesicle in regulated exocytosis is secretory vesicle. It occurs commonly in secretory cells and not in all cell types. It has temporary or key-center-on fusion. I want to talk about regulated exocytosis steps. Regulated exocytosis has all steps of constitutive exocytosis, but one step more. The first step is vesicle trafficking. The second step is tethering. The third step is docking. The next step of regulated exocytosis is priming. This step is only for regulated exocytosis. There are modifications in some of the cell membrane elements for exocytosis to take place. Secretory proteins are released only when triggered by extracellular signals. And the final step is temporary or key-center-on fusing. The vesicle temporarily fuses with the cell membrane for a period of time to allow the formation of the fusion pool for releasing the cell contents to the exterior of the cell. The vesicle then detached from the cell membrane, where it reforms and returns to the cell cytoplasm. You can see all steps of regulated exocytosis together. The third subgroup of exocytosis is lysosome-mediated exocytosis. This process involves the fusion of cell vesicles with the cell lysosomes. Enzymes contained in the lysosome breaks down the molecules and carries this waste material into the cell membrane, where it fuses and releases their contents into the extracellular matrix. Material transported by lysosome-mediated exocytosis are waste materials, microbes, and cellular debris. You can see all steps of lysosome-mediated exocytosis here. Thank you for watching. Now it's time for the quiz game. I have put the link to the quiz game in the description of this video. Download it for free and enjoy it. Please give me a comment about the quiz game. In the next video, I will talk about another biomedical topics in a simple, classified, and summarized form. Until the next video, bye!